talk about something a little bit different for this section, I think, and talk about disease resistance in honeybees, and specifically the social conditions where disease resistance is really important. So one of the things that really excites me about honeybees and social insects in general is this interplay between individuals and the colony. So colonies are made up of tens of thousands of individual honeybees that are all non-reproductive. There's one reproductive queen. And so the colony has to survive, and if those colonies don't survive, then all those individuals are lost. And this becomes particularly important when we're talking about immunity. Now, I think when we first think about mechanisms of immunity, we think about individual immunity, partially because we're individuals ourselves. And so these are things that are common across really all organisms. And the first line of defense are physical characteristics, so like the bee's cuticle, preventing parasites from even entering the bee. But then there's a host of physiological mechanisms, like the production of antimicrobial peptides that I'll talk about a little bit later. But now the other aspect here is, again, thinking of colonies as kind of even a superorganism. And honeybees have, and other social insects have what we call social immunity. And these are individual traits and behaviors that effectively reduce disease transmission at the colony level. And I'm going to get into specific examples. So one that loosely fits into this idea of social immunity is multiple mating of the queen. So queens go out and fly out early in their life. They mate with, on average, 12 males. And from a disease perspective, multiple mating increases that genetic diversity within a colony, and this has been shown to reduce disease prevalence, but also intensity of infections. Now, there's a lot of other non-mutually exclusive reasons why queens might multiply mate, but we're focused on the disease aspect here. And so this is really a passive effect. A colony can't change the level of genetic diversity that they have within that colony. It's kind of this background level effect that is having these impacts on disease resistance. Now, one other behavior that I've studied a lot is the use of antimicrobial plant resins by honeybees. So honeybees collect plant resins, like you can see here, dripping from this pine tree on a wound, and they also get it from leaf buds. These are highly antimicrobial. Bees collect these resins, bring them back, and incorporate them into their next architecture. And this is a genetic trait. So how is this social immunity? So previous work has shown that when colonies are enriched with resins, it knocks down the general bacteria in these colonies, and the immune system of those bees within those colonies is basically relaxed, so it frees their immune system up to fight off pathogens. It is also has direct effects against a larval fungal disease called chalk brood, and they actually self-medicate with resin. So if you give them this fungal disease, they will increase resin collection. And so in this way, it's both a prophylactic or passive effect, because it has these background effects in the hive on immune systems, but it also is an active effect. They can bring in more resin when they're challenged with a pathogen. Probably the best studied aspect of social immunity in all of the social insects really is hygienic behavior. Now, I know that's a general sounding term, but it's a term for a very specific behavior in honeybees. So it's essentially the detection and removal of disease larvae before it becomes infectious. So it doesn't necessarily protect individual larvae from disease, but it can eliminate the disease at the colony level by removing those individuals before they become pathogenic. And this is also another genetic trait that has been bred for quite significantly over the years. And then again, this would be more of an active effect, so it's something that if a disease is presented, the bees will respond to. And this is also effective against chalk brood, that fungal pathogen, among others, bacterial and mite parasites. So the big question here that I'm tackling, and it's a pretty big question, is since honeybees have multiple defenses, and they use multiple defenses, and a lot of these defenses can be active against the same parasites, how do they invest in these defenses? what's the evolution of basically parasite disease resistance in honeybees, and are there trade-offs between these different defenses? So in order to tackle this, I set up a series of colonies that were headed by sister queens and that had undergone restricted natural mating so that we could have a variation in genetic diversity. And this genetic diversity was determined by genotyping offspring. So I ended up setting up 15 what I'm calling low diversity colonies, and these were headed by queens that had mated with 4 to 7 males, and then another 15 colonies headed by queens that mated with 14 to 30 males. And then I looked at the effect of genetic diversity on disease resistance, and then also resin foraging, so again, both the prophylactic or background or passive level use of resin, but then also how they respond to a pathogen challenge, and hygienic behavior, and then I looked at larval immunity, and then I'm not going to be able to talk about it, but I also looked at grooming. So in terms of the role that genetic diversity played in disease resistance in these bees. So again, the low diversity colonies here were on average 4 to 7. The queens were mated with 4 to 7 males. The high was 14 to 30. Here's really just the proportion of in levels of infection. So these are the number of mummies. So these colonies were given chalk brood, which turns larvae into these chalk-like mummies. And so these are the number that were found in the colony after a period of infection. And you can see this isn't significant, but it's suggestive that in these low diversity colonies, there is an increased, we do have some that have high infections, and there's more variable levels of infections in these low diversity colonies, which is supporting what previous studies have shown, showing higher intensities and more variability in these low diversity colonies. However, all the previous work that had been done had pretty much been done on single drone inseminated queens versus ones that were inseminated with 12 drones. And so here I'm looking at a little bit more intermediate levels of diversity. So it's suggestive but showing that genetic diversity at these moderate levels isn't quite as significant. All right, now in terms of resin use. So in order to determine how much resin the bees are bringing in, basically close up the entrances to hives, and we look for the number of returning resin foragers. And here, this is a pollen forager, this is a resin forager. I've had lots of practice seeing hundreds of these, so I can tell the differences, but you might not be able to from this one picture. But basically, we close up hives and we count the number of returning foragers over a period of time. We did this for about 2 weeks. And then I challenge colonies, and then we count again. So we have kind of this pre-challenge baseline level, and then their response to the fungal pathogen chalk brood. In terms of how genetic diversity affected this, there's no significant difference in the total number of foragers collected during the pre-challenge or prophylactic period. However, there's increased variance in these high diversity colonies. So this is a trait where it's very, very few bees are collecting resins in the hive, about 1% of the foragers. So it's a really small number of bees that are performing this behavior. At these sample sizes, it's not wholly unexpected that we would see higher diversity, the higher diversity colonies having higher variance there. But we did confirm that honeybees in North Carolina also self-medicate. The previous work was all done in Minnesota. Here's the total number of resin foragers, and then we have in the pre-challenge and then the post-challenge period. Each line indicates a different colony, and the arrow in the middle is the average. So you can see that there's a lot of variation in this behavior across colonies, but overall there was an increase in resin collection. And this was started just 5 days after this pathogen was introduced, so it's a pretty quick and immediate response. Now, hygienic behavior, the next social immunity. So there's a really standard way to assess hygienic behavior in honeybees. Basically, you freeze a section of comb that has developing pupa in it with liquid nitrogen, and then 24 hours later, you go back and you look and see how many pupa they removed that have been dead. So hygienic behavior is on a spectrum from 0 to 100%. This would be a very hygienic behavior because it's removed all of those dead pupa. This one would not be very hygienic. You can see they're starting to remove some of them. Some of these they'll never remove. So in terms of genetic diversity, so here's the percent removal at 24 hours. We have the low diversity again, the high diversity. You can see there's no real strong difference here in terms of genetic diversity, again at these levels. And again, these are sister queens. Hygienic behavior is a multi-locus trait, so we probably need larger sample sizes and more diverse samples to get at this for sure. In terms of hygienic behavior and residues, so here we have the pre-challenge or prophylactic residues, and then here we have their responsive collection in response to that pathogen. See, there's no relationship here between hygienic behavior and residues. All right, now to bring in the individual immunity. So what we did here was we studied the immune response of larvae to a pathogenic bacteria. So larvae were taken into the lab, reared in 96-well plates, and then treated with this bacteria. And if we let these larvae go longer, this bacteria essentially turns them into this brown, smelly goo. But we collected them 24 hours after infection, where the most upregulation of antimicrobial peptides happened. Then we stored these larvae and did real-time PCR, looking for specifically this antimicrobial peptide hymenoptacin. And expression of these antimicrobial peptides not only is shown to resist infection, but it's also a genetic trait, so it's something that does have some heritability. So in terms of some quick results here, basically the analysis that I'm showing here and talking about is the difference between control larvae and AFB-infected larvae within a single colony. And we didn't find any difference to genetic diversity in the colonies for this study. In a larger study, we did show a trend related to genetic diversity, and there was no relationship in terms of individual immunity and resin collection. However, here's the one positive finding for you guys today. So we did find that larvae collected from more hygienic colonies had a higher immune response. So larvae from really hygienic colonies have a stronger immune response. And hygienic behavior works against the same disease that we were giving the larvae. So it's kind of interesting that they might be additive effects there. And really the question here then is, are they latent traits? What's going on here? And so there has been some QTL mapping done for hygienic behavior, but most of those genes identified are involved in odor and binding and olfactory processing, because those hygienic bees are able to smell sick larvae. And so there wasn't anything totally suggestive there in terms of immune pathways. However, some recent proteomic analyses have found that larvae from selected hygienic strains have increased levels of this peptidoglycan recognition protein. And so higher levels of this protein might allow for a larva to increase activation of its immune system more effectively, which then would increase their immune response. And so the question really here is this kind of a predictor or initiator of future hygienic behavior. But so we need to really look at this a lot more, I think. But just to wrap up quickly, we really didn't find any trade-offs between these behaviors, and there really seem to be complementary and non-additive effects in social immunity. Resin use is clearly self-medication, at least in response to chalk brood. And genetic diversity has kind of more modest effects at these moderate levels that we're looking at. Now, again, the big question here now is, are hygienic behavior and larval immunity and adult immunity correlated and additive? And so we need to bring in the grooming results which also doesn't appear to have any relationship with these other social immunity mechanisms, but maybe genetic diversity. But the bottom line is, this is all helping to help see which traits we can promote for honeybee health, and now that we know there aren't any trade-offs, we could breed lines that would resolve these behaviors effectively. So thanks for sticking it out, and I'll just take questions up here at the end if you want.